All right. I'm pretty sure we're live. We're rolling. Let me see. It looks like my audio is doing the little bar thing. Yeah, the flow hood cooling down some substrate. Still warm. It's still pretty warm. So you might hear a little flow hood buzz in the back, but somebody's got to cool the substrate. So little delay. I was trying not to have to use my camera because it is a nice camera, but it still comes through pretty on the live stream. So I was trying to use just my computer camera to maybe help it all keep up with itself, but it's not doing it. So we're gonna rock with this. And yeah, today we are going to be talking about preserving wild species, cloning wild specimen, running wilds, and just kind of why wild genetics, wild source genetics should be preserved, and I think should be run in a lot more farms around the U.S. Beyond just, let me share this. Beyond just running local species and the sustainability aspects of running like an oyster that you found from up the road. And long term, those spores aren't going to be any issue because it's already kind of been found naturally occurring, unless maybe a gold oyster. But there's a beauty in being able to find local species that are thriving and growing really well in your local environment, in your local conditions, and ecosystem, and being able to take those and convert that into one of the cultivars that you run at a farm. So then all the spores that you're kicking out from your grow room, all of these things are from a local species. So we want to make sure, especially when we're producing at a huge scale, not to bring in like invasive species and things like that. They're going to outcompete our wilds. There's a bit about golden oysters and that's a video for a whole nother day. But today, I just want to talk about going out in the woods, finding cool species. My experience with wild cultivars so far, whether I found them locally or sourced them from friends, that's just as cool, just as important, is getting them from some friends, and just being able to save those genetics, whether it's you or other people, so that long term we don't lose them. So let's see, Google Slides. And seriously, I gotta do some more research. But if anyone's got the live stream, details, I can do this, I can do that, tips, tricks, let me know. I gotta do some diving to improve some things, but make things a little smoother. But that's not what's really important. I got my name on here. Can do the thing. Can do the wait. Yeah, that ain't bad. I think I can move myself. I don't know. That's fine. Last time I went without myself, and then I realized I was making some faces and had some good facial expressions that probably would have added to some of my points. But today we're in the swamp. Block's cooling. Talking about preserving wild specimen. Mostly why. I'll give a little bit of tips on cloning, and kind of how I do some things. But I just kind of want to share my experience with some wild cultures and why I tend to work with them. And so actually this picture here isn't specifically mentioned in the slideshow, but this is I mentioned my bear's head. This is a main bear's head, but this is the HAOG2. So it was the second different bear's head specimen I collected within a trail on the woods. And this one was probably about 75 feet downstream from the other one that I found the previous year. But the previous year collection is like a way different organism. They grow really similar on agar, but fruiting, and this one just does what it wants. So it's kind of awesome. But that's also part of the fun of wilds, is you get some different representations of different things. So while they're all lion's mane or all bear's head or all oysters or something, they're not going to all be the same. Like this first one I want to bring up, I'm just gonna have I have a couple different like my wild cultures that are not my specific, but the ones that I've run. Mentioned who I've gotten them from, and where they come from. And so I just want to talk a little bit about those, my experiences with them, and why I think that's part of what's so special about these wild cultures. A background, I guess. I'm down here in Jacksonville, Florida. It's hot. It's August 24th in middle of summer. It's been 95. The cicadas have been ripping all day. The Instagram things when people are posting right now, and some different sounds that you just hear of like, oh, it's that hot. The guy hitting the harmonica. That's how hot it's been outside. And despite this, mostly with some wild cultures, I've been able to have still lion's mane fruits throughout the summer. It's not always ideal. I'm not going to say you should open up a full massive scale operation fruiting lion's mane at 95 degrees, but it can be done. So that's what I'm doing. Is I'm out here. I'm growing small scale. Trying to see what works, what doesn't. Trying to focus on education and community, and running a bunch of other people's wild genetics, and going out and finding my own wild genetics to preserve those and kind of intermingle those, and kind of find some more heat tolerant species. So one of the cool ones, and the ones I'm most excited, and what kind of sparked my idea of doing this live specifically, was the black marsh lion's mane. And so this one, it's a lion's mane. It's Hericium erinaceus. There's some talk about lion's mane, some studies and papers about lion's mane possibly, or Hericium erinaceus possibly being broken up into like 4 or 5 different species, like Hericium carolinense, organense, 2 or I don't remember the others. Hericium erinaceus is lion's mane to me. I'm not changing it yet. So the black marsh lion's mane, this comes from Maryland, from the Black Marsh, something called a marsh called Black Marsh in Maryland, and it was found by Mycosapiens on Instagram. If you haven't followed them, follow him. Follow Forager's Folly. If you're in the Baltimore, Maryland area, anywhere around there, really check him out. He's got a lot of really incredible native plants, as well as kind of working on the intersection between plants and fungus, and different ferments and feeds. Really cool IPM tips, all sorts of really good knowledge. So for more than just fungus, follow Forager's Folly. But Mycosapiens, that's the dude. I've got a couple other cool wild cultures from him, including a wild Maryland beefsteak. I've tried the fruit a couple times, but it doesn't really like the heat. So we're still working on it. And then I've got a wild golden gnocchi that I've gotten from him as well. And so all of these get backed up. But the point here is that this black marsh lion's mane, I was my most recent couple flushes, and I collected spores off of it, so that way I can enter it into my breeding program. Just getting started, isolating some different monos from each of these wilds, to be able to preserve them as well as their genetics and move them forward. And so this black marsh lion's mane from Mycosapiens, I've had success fruiting it 95 degrees, probably down to like 40-50% humidity. We're starting to get rain daily, so most afternoons we'll get about 30 minutes of rain right as it's about 95, 96, 97 degrees Fahrenheit outside. Pours rain for 30 minutes to an hour. Pours, especially being used to the Maine rain for a while. Now that I'm back here in Florida, Maine, like a pouring day, a day of just like absolute, or any downpour in Maine is like the slightest sprinkle in Florida. It's crazy. So we're getting that daily. So that's definitely a nice buffer. The afternoon rains will be able to bump up a little bit. Humidity kind of keeps my tents damp for a little bit, but they're still going to dry out. So this black marsh lion's mane has been able to handle that like 95, 96, 97 degrees and down to probably pretty low for 30-40% humidity. Those tents run pretty dry pretty quick. I had a hygrometer out there, but they keep breaking, so I'm not going to keep buying new ones. I'm just going to keep watching it. I need to get some of the low scale or low tech, just wall circle, Old Faithful hygrometers, temperature wise, even just to put out there. But I'm broke, so I don't feel like buying one yet. So I'm just reading the mushrooms and reading the weather app, and feels like have been about 115, 117. So this thing's still throwing down. This harvest was just a couple weeks ago, or 2 weeks ago, last week, 2 weeks ago, last week. I think it was like last Sunday. I don't know my days anymore. Every day is just different, but the same. Whoever made up days was a fraud. So it's a super nice, beautiful, real dense, long teeth. I top fruited one, and the top fruit was really nice, really dense, coming in well. But then it started to lose some moisture because it was top fruited. So that was just a big open surface. But this side fruit here, and I had another smaller block that I side fruited. And just nice, like you can see, really nicely broken up. It's not huge mains. It had really nice long teeth for each. So this one's a really nice one to be able to actually break apart and get into little baskets and different assortments compared to other Lions mains, where maybe you're just getting one huge main or a couple big mains. This one seems to like to throw a couple smaller little segments in there. And it's still a really nice dense species. So again, that one I collected some spores off of. And I'll have a picture later on of my Lion's Mane Spore Tech. And actually this I got from Mossy Creek in one of his live streams. And so if you haven't checked those out, the Mossy Creek live streams, he's got a lot of great information hidden in there. But to save Lion's Mane, I would take one of those chunks off that Lion's Mane, one of those mains. And the best way to get spores, because with all those teeth, if you just set it right on aluminum foil, that pom-pom right on the aluminum foil, it's going to get a little more bacterial. There's too much surface area. They're too wet, too fragile. So what you want to do, you can use a smaller mason. This is just a mason jar ring. So you can use even a smaller mason jar ring, soak it with ISO, get a really good clean. You can sterilize them if you want, but I haven't found the need to do that. Just a really good ISO soak, rub, let that dry off, and then you can use the mason jar ring and put those mains on top, and then that gives them that open space here to sit above the aluminum foil. And so then that's how you collect really good Lion's Mane spores, is just using that mason jar ring as a really nice little riser. Us mycologists, we have mason jar rings up the ass, like they're everywhere. I have so many of these. The smaller ones usually work a little bit better, but either way, that's the best way to get Lion's Mane spores. I'll talk about that later, but I figured while I'm talking about it. So this one was nice to collect little spores off of, just to be able to rip off a bunch of those little tiny manes. They were each nice, dense. It's kind of had like a nice sweet smell. Some Lion's Mane's a little sweet, others, absolutely. Mossy Creek, I've learned a lot. So much information has just been from Mossy Creek, just going in my headphones in the background. I've watched so many videos over the years. So if you haven't, go check out Mossy Creek. He's got literal days upon days upon days of YouTube videos, live streams, focused content, a lot of different information over the years. So he's a great YouTube account to check out if you haven't yet. And yes, this is also why you should clone wild genetics more than anything. It's because life is just damn beautiful, and nature does it better than most people can. So Black Marsh Lion's Mane, it's beautiful, it's really cool, it's awesome. It's a little smaller font than I'd like at the bottom, but you can see, so in talking to Mycosapiens, where I got this culture from, in Maryland, they're facing a lot of the beech leaf disease there. And so that's wiping out, it's, I guess, spread through, I think, like nematodes or something in the water. And then it wipes out a bunch of the beech trees. And so that was, I think, where this Lion's Mane came off of, and also where a lot of Lion's Mane are found in the area. And so in the future, we're going to continue as the beech leaf disease continues to progress and be a thing. We're going to continue to lose more trees and possibly lose a whole lot of biodiversity in that region. So that's even one of the reasons why the black marsh lion's mane, not just it's really cool, it's beautiful, it has really nice manes, like smells good, tastes good. Yeah, it's a great culture. But it's also important because it's one of the cultures out of an area where we may not see as many of these specimen and a major loss of diversity in these specimen over time. So that's one reason why we keep the black marsh culture going. So we're going to work with that. And it was great talking to Mycosapiens just about that and just kind of get a little bit of a perspective on working with and saving some of the wild cultures, whether they're from my local area or like this case where they're from Maryland. You're working with some wild cultures that your friends send you. Sharing is caring. So this next one, June cut. This one's a beast. This one's crazy. So in this one, it's got a couple smaller manes, but that's a top fruit as well, so they are bigger. It does tend to throw some bigger manes, but this is a Pennsylvania wild lion's mane, one that is outperforms every commercial variety I've run next to it. Not a bunch, like just a handful of commercials, but a couple different commercial varieties and run in comparison. This thing in the outdoors, in the heat, with no technology, low humidity, has been able to outperform just about every culture of lion's mane I've ever run, I think. And it's been able to do it, produce really long, nice, really long teeth. I've posted about this one a lot on my Instagram, and it comes from Uncle Fungus. And I didn't find it myself. I purchased this culture from Uncle Fungus, and I've been running it for a little while. He noted it as heat tolerant. I think found it on like a 90-degree day or something in Pennsylvania. And so I've been able to run it at like 96 degrees plus in the tent down here in Florida. So even more of an intense heat and intense UVs. And I just use a little shade cloth over the top of some plastic. Again, check out if you haven't seen it. I post about it all the time on Instagram. So check that out, or if you don't see it, I'll probably post about it again soon, or check out the Discord. And I posted a little bit about my setup in there. But really basic setup, and this lion's mane culture, it's a really fast turnaround. You'll see some lion's manes on different agar recipes are a little slow and just kind of a little finicky. This one just rips. And even if it's being a little slow on agar, as soon as it hits grain, takes off, and it'll colonize grain. Whether it's a jar or a decent sized bag, it seems to take about 10-12 days to colonize grain. Maybe about 2 weeks to colonize grain. And then as soon as it hits substrate, it's probably about 10 days, and you can slice the bag open. A little bit longer, probably for some people and places. It is sitting in about a 75-degree incubation here. It's pretty warm, so speeds it up a little bit for sure. But then it's able to produce some really nice specimen. So this one is a beauty. I've been really enjoying it. And it's just one of the fastest, most aggressive cultures I've had. And it's been able to fruit outdoors in the heat when the cicadas are just absolutely ripping and chirping. So I've got a good run of it coming up soon. I've got a couple jars colonizing, a couple master jars colonizing. I'll put in some grain bags and try and run a couple more bigger numbers of this, of the June cut. But this one's been crazy. It's been great, and it's coming from Pennsylvania. And so there's all sorts of different pest disease things all over the place. And so while this one's from Pennsylvania and I'm from Florida, it's still something I've backed up. I've taken spores of, put in water culture, and I'm going to work with it further because it's still from an area and from somewhere we don't know that we're always going to have access to these genetics. So just because it's not immediately threatened, as far as I know, doesn't make it less special, right? And that's what, right? Yeah, absolute beast in the heat. Want to make sure what that little one was. This one, Virginia Lion's Mane. I got this one, another Hericium erinaceus. I've got more Hericium erinaceus wild cultures than anything else. I've actually got this one. I just shook. It was just starting to go. I just shook it yesterday. You can't really see it at all. It's lion's mane, but that's a Mongolian lion's mane. So I've got the Mongolian lion's mane, which I'm really excited about. It's not this one, but Jacob at the Mushroom Shop LLC. Full name every time. Full respect. Shout out to Jacob. Mushroom Shop, the Mushroom Shop LLC. He's awesome. Incredible cultures. And so he shared the Mongolian lion's mane with me. And so yeah, that one's got some heat tolerance as well. So I'm excited to run and work with that one. But anyways, the Virginia lion's mane, what I have posted here. I don't mention all of my live cultures, just the ones I've been able to run a handful of times here, just so we can talk and have some examples of what some different wild lion's manes, different wild genetics can do. This isn't a one-stop shop, everything wild here. Just kind of providing some more inspiration, some ideas, and just showing my experiences with the wild cultures I've worked with and run. And so any questions, anything that comes up, any of your experiences you want to share at any point, obviously feel free to throw those in the comments. But the Virginia lion's mane, this one is about not as heat tolerant as the black marsh, and not as heat and drought tolerant as the black marsh and the June cut have been. This one I got from Lion's Mane James, Performance Fungi, on Instagram. If you haven't tried Performance Fungi and their extracts, go get yourself juiced. Tell them I sent you. Don't tell them I sent you. Either way, they got the best extracts. I got a bunch over there. And just take your mushrooms, right? If you guys stop the video and you can get that little QR code, right? If you stop the video and you can get that QR code on these stickers he's got, you can save money on extracts. So don't say I never did nothing for you. Or I don't know. It's probably blurry. I don't know if it's gonna work. I tried. But take your mushrooms. Performance Fungi, best extracts in the game. Really great extract, especially lion's mane, cordyceps, those big hitters. He's also a great dude. Lion's Mane James is his Instagram, and so that's where I got this culture from. It's from Virginia, and it's got medium heat tolerance, right? I'm not gonna sit here and tell you all of them are fucking bangers, top of the notch, no matter what the temperature is, just because they're wild. They have a lot more tolerance and a lot more leeway than a bunch of other cultures because they're wild and they haven't been babied. But even still, this one, it did pretty well. I fruited it indoors in the tub as well because I was kind of struggling outdoors. But it's got it was able to fruit in the mid 90s, but I think really this one's looking at probably like an 85 to 90 degree max out for really good potential, really, especially down here in Florida. And also, if I could maintain a higher humidity, I think that would go a long ways. But instead, I've been hands off, just kind of letting them run outside underneath that shade cloth. So this one did pretty well. Still better than a bunch of other lion's manes that I've put out there in cultures and blocks that have literally just died in the heat overnight. So even its ability to try and push out some fruits was pretty well, and it was able to put out a couple nice little pom-poms. The top got a little bit dried out, so that's maybe why I'm not showing the whole block. But it still was able to push out some nice pom-poms outdoors and in the heat. And so I got some spores off of that one as well. And it had a nice sweet smell to it. It wasn't quite as dense as some of the others, but it was nice and sweet compared to some of the other lion's manes that are sweet, but like mushroomy. They're lion's maney, and they kind of have like a almost like Hericiums almost have like a chalky something smell to it. It's like a smell that has almost like a texture behind it. I don't know if anybody else feels that. There's certain smells. It's one of my we'll do a video. We'll do a video one day about the smells in cultivation or the smells of fungi or something because yeah, I love it. And so it smells really, really nice. But also, this one is one of the really more aggressive myceliums that I have. So it's really fast to colonize, but also on plates, it's fast to colonize, and it's always got one or two offshoots. Lion's manes, you'll see it's kind of hard. They'll want to fruit or they'll want to be in vegetative state, but once they're fruiting, they don't always want to go back to vegging. And so this one's kind of a little bit in that, and it always just has one or two fruits. It's always creeping through like the bottom of an agar dish, right? Like, I don't have one right here, but it'll creep through the things here, and then I'll just see a little pom pom on the bottom of the dish. This culture does it more than any culture. Um, and so that's just one of those things where it's another one of these. Each culture, especially each culture in cultivation, even commercial cultivation, it's its own being, right? They act differently, they behave differently. But what I've found is really these wild cultures have just more character. You know, they're just more different, which isn't a good way to say it. But they're just their own beings. They're fun to work with compared to some of these others. They're all just very different. Whether it's on the agar stage, the spawn stage, the substrates, or of course the fruits and their potential to handle things. So this one still shows a lot of potential. I'm excited about that one. The next one, Brick House Lion's Mane. So I've run this one. I tried to collect spores, but I didn't it didn't give me any spores, so I got to run it back. But this one's cool because this one comes from in Washington D.C. street side, collected by Microdex. If you go back on his page on Instagram, you can find it. But shout out Microdex. If you don't follow Microdex, he does a bunch of really, really, really cool stuff, kind of at the intersection of food, fungus, and bugs. He's not eating the bugs yet, but he might soon. I don't know. I'm trying to talk him into it. Some of the little larvae just look like little scrimps. So if you don't know what I'm talking about, go check out Microdex on Instagram or YouTube, and you'll know what I'm talking about by these little scrimps. But this culture comes from him. It's from DC. Obviously, in DC, there's a lot of pollution. There's a lot of urban sprawl. There's not much green space. So being able to even just have some cultures from that area are neat because those have kind of proven to be able to handle a little bit of that human disturbance and some of the human pollution and things like that, and still be able to fruit and live their life and do their thing. And so this one. It's a slower. It's got nice dense fruits, but it's a slower specimen, and it hasn't been since day one. It's not as heat tolerant. It doesn't really like it too hot. Really, it's done way better if I can keep it indoors, so like it's under 80 degrees. But it's still really nice and dense. And in like a grow room, in an actual controlled grow room, it performs pretty well. But it's a slower colonizer, and the mycelium's a little bit slow. And so talking to Microdex, he was saying about how he thinks it's also possible. It's just a little bit of an older specimen, which is also I wanted to bring up and show this one off because I don't know that it's an older specimen, but I've seen in a few different research papers and books and things, it's been noted, and I'm not sure exactly how they come up with it, but it has been shown that the lion's mane in the wild, lion's mane usually will fruit, or not usually, but has been seen to fruit off of logs and trees in the wild. One single specimen, one tree or one log. Maybe it's some spores taken over, but for up to 20 years. And I guess the scientists have been think it's possible that the lion's mane, given the circumstances in the wild, with the right log, it has a big enough log or right environment or something, that it could live for up to 40 years. Which one lion's mane living in the wild for 40 years, 20 years, that's pretty impressive. No, I mean, like, that's wild. That a mushroom, lion's mane, as delicate and soft as that species is, obviously that's the fruit body that we're talking about, not the mycelium hidden beneath the log. But I should have put the source on here. But look it up. Maybe I'm bullshitting and I'm looking in the wrong books, but I've seen it a couple times, and it's crazy, basically, just the diversity and how long some things can live. And so this one, maybe it's an older specimen. And so if so, it's an older specimen that's been living in DC in this urban environment, buildings, houses, cars everywhere. And so this one that I need to grow back out so that way I can take those spores and then keep this specimen around long term. Next, we're gonna change it up off the lion's mane. Finally, I've got a couple others. All right. So this one, you can see it's really the textures on it are cool. The bottom right picture is the wild collection itself. A couple days before I collected it. The top right picture is this specimen fruiting down here in Florida this spring, probably mid 80s. It topped out about mid 80s, but it was getting a little bit cooler, still to like low 70s at night, mid 60s at night. And then the picture on the left is where it came from a grow room. I had actually kind of top fruited that one, but it's Maine Bear's Head, Hericium americanum. This one actually, I've talked about it, I think, in a couple videos. I've talked a little bit about it, but the patch of woods that this species came from has such a soft spot in my heart. I miss going to those trails. I didn't know how good the fungus in that old growth was until I was removed from that old growth forest, and now, especially now here in Florida, it hurts. It was crazy. So it was in Western Maine. I'm not going to be too specific. It was Ordway Grove was the spot, but it was in Western Maine. And it was this really old growth forest where, looking into it, like the history of it, I guess, like Civil War generals and artists and poets and things for years and years and years have come up to this spot in the woods just for the tranquility, for the massive pine trees, and some really big oaks, some just really big trees, a lot of big pines, really big pines. But a lot of just huge old trees, and I guess people come up there forever. It was in private property, it's entire like private ownership, and so now it's just an old growth forest. And so I would go there every day, and there I found two bear's head cultures, a lion's mane that I lost, and it hurts. But you could tell it had the deer butt shape, right, and rather in this shape. And so, but there were chanterelles, black trumpets, boletes, amanitas, all different Lactarius, russulas, this, that. I found a couple chicken of the woods flushes I ate. I found some matsutakis. It was rishis. It was crazy. It was like a 20-minute walk, 20-minute loop in the forest. So that's where I got this bad boy. And it's probably mid 80s is where it peaks out. I found it in August in Maine, so it was warm but cooler, right? Starting to cool down about this time of year. In Maine, in the deep woods, so definitely a little bit cooler than down here. It likes medium temperature, humidity, but it's got a really, really nice, like licoricey type smell in the best way possible. Like I don't like licorice or black licorice, but it kind of has a little bit of that to the smell, and just like a nice mushroomy, a little chalky, but like also flowery on the smell. It's a really interesting, the mycelium and the fruit itself smells and taste. It's like a way more delicate, even texture wise, but just flavor and taste. It's a softer, more delicate lion's mane. It's really interesting. It's got coralosins, which is its own host of compounds that we're not going to get into, but possibly very, 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 very potentially beneficial as well. But then the mycelium on this one, so like some of the lion's manes, especially on an agar dish, they're a little picky, a little finicky, or can grow a little bit slower. But the bear's head, these like almost keep up my oysters, and they don't really care what the media is. They're just gonna grow. So it's a really awesome culture. I should have put the agar up. It looks really awesome. But this one came from the old growth forest. It's been around forever. I've got some spore prints of it. I've got it cloned in water vials. And while I don't think those woods are going to be changing hands to anybody, it's still it's possible that somebody comes in, buys it, tears some trees down, some different pest or disease come down, whatever it may be. But I've at least now got these genetics from this untouched, pure old growth forest, backstocked and preserved. And they're nice. They're not maybe not as vigorous in the heat, but they're also from Maine. And so I'm excited for this winter to get this one rolling a little bit more. Collect some spores, run another generation or two off of it, or run the spores I have, and maybe even find some other wild bears heads from some other people's to be able to intermingle some of those genetics. But this one's really cool, really sweet, delicious, soft, delicate lion's mane, basically. But the mycelium is like extra vigorous lion's mane. And you can't really see it again because it's a lion's mane. But I just shook these the other day, and so you can see the lion's mane-esque, real light, wispy mycelium. If you can see that, sorry, the light's bad. But that's the bear's head. So I'm gonna try it again. It's 95 degrees out, but somebody's got to try it, right? Somebody's got to push the limits. And worst case scenario, it dies in the bag, and I give it to the garden. My wallet doesn't love that, but also, yeah, somebody's got to push these genetics and see what we can actually accomplish. So the Maine bear's head doesn't really like the heat as much, but it's still a really cool, really beautiful, unique specimen from a nice old growth untouched forest. So this bad boy is pristine. This is definitely what Mother Nature selected. And then this one, I should have grabbed the plate. I meant to. No, all right. I'm not gonna do it because I couldn't immediately find it. But this one, I'm excited. This is my first Florida clone. The Ganoderma courtesy. I always want to say courtesy, but courtesy. It's a golden reishi, is technically what this is called. And so I found this in a trail not that far from where I'm at, surrounded by suburbs. So it's just one little patch of kind of like a pine, palm, flatlandy kind of spot, surrounded by suburbs everywhere. There's soccer fields on one side of the park, basically. And within that, I've seen a couple now, but a handful of weeks ago. But Ganoderma courtesy. So this one is my first Florida clone. So I'm excited. And it's got a really cool, dense mycelium. It kind of produces a few like that gold color in the center of the cap. It produces like some metabolites, kind of that color, and it's not as like uniform as a bunch of reishi grows out in like a perfect uniform disc. But this one, kind of the cultures I've got of this species seem a little kind of off and a little elongated here and a little shorter here, and they kind of just have their own. They're slower compared to a bunch of other Ganodermas. Probably about a week slower, maybe even more, 10 days slower to colonize a dish, and even probably about a week slower to colonize grain than like a sessile that I just was running. So this one, I'm excited. This one is just hitting grain. I've got a same culture or same species from One Holy Ghost at One Holy Ghost on Instagram. I've got a Ganoderma courtesy from him that he got from. I've got the iNaturalist for it, the iNaturalist ID, but it's I believe from Boston or the Boston, Massachusetts area, and it's a Ganoderma courtesy. So I'm gonna collect some spores from that. I'm gonna collect some spores from this one. It should be hitting grain the end of this week, so I'm excited for that. And then I'll have some different spores from some Ganodermas, and maybe I'll, make them kiss. I will. I might not. I keep trying to. I want to do it with everything, and at least just see what crossing, wild with wild can get me in terms of. Really, more than anything, I'm just trying to build resilient cultures and like easy cultures. I want them to look really nice, be really beautiful, and have good like compounds and things within them. But more than anything, I just want to grow cultures in not less than ideal, but like less than ideal ways. So that way, when I give people, I teach people how to grow, or I give them a different like grow kit or ready to fruit block. They can actually put it on the counter or in their closet or somewhere that's not that nice, but it'll. Some of them still perform great. Don't get me wrong. A lot of them do really well. But I want to, especially down here in Florida, where it's so hot. It's hard for a lot of people with so much heat and so much cement everywhere to grow things. So that's really part of my goal, is to be able to take all these wild specimen from all these places, as well as a bunch from down here, and just keep collecting them and keep working them and find what people can actually run and work with and produce down here in these conditions, whether they're beginners or trying to actually start up a farm. So that's why I'm doing it all. But this one, first Florida clone. So hitting grain soon. I'm excited for these. If you haven't seen or found any of these, the courtesy in the wild, they've got like a small little antlers, like nothing. They're not like huge conch growing out of a tree or something, but you can see the color, nice little antler on that. Just waiting to be nice reishi art. So I've actually run them in a couple. I just used the last two today to spawn the other courtesy. But I've got a bunch of those little like Tractor Supply buckets, or like mini 5-gallon buckets. So I'm going to grow a couple of reishi in some of those. And I also inoculated or filled an empty garam marsala, a little glass spice thing. I put a couple of the Massachusetts Scanoderma courtesy in there. So just today, a couple hours ago. So I don't got nothing to show you guys. But come back in a couple weeks or a couple months, and that thing will probably be fruited. So I'm excited for the courtesy. Courtesy. I don't know why my brain can't accept that. Especially the Florida culture. So I can get into some heat and some cool reishi art in the heat. And I think those would be cool to kind of get people excited and show people some of the different potential, whether it's at some different classes, at a different event, setting up a stand, something eye-catching, and just different. So this one coming soon. This one I'm excited about. I'm a cultivator before anything else, right? I like to learn about my surroundings. I want to learn my local fungi. I want to work with them, but more than anything, I'm a cultivator. So, obviously, I love lion's mane. I love oysters. I love reishi, all these classics, these basics, everything that we're used to. Run all of those, save all of those. You see wild lion's mane, wild oysters, any of these wilds, save them. But sometimes, some mushrooms just kind of stick out to you a little bit, and something about them. The colors on this one, just bright yellow cap. You can see the baby on the left. On the right picture is a small little mushroom and a medium mushroom. And then on the left, I cut it in half. And so this is a Butch Waldo. It rolls right off the tongue. Whoever came up with this name, I have another one I should have mentioned, but I can't even think of the name. But this one is Butch Waldo Boletus hemicrisis. Easy, right? You probably you didn't even forget it. It's locked in already. But this one I actually found in the same area that I found that golden reishi. And so what's cool about this one is it is a saprophytic mushroom, and it is also mycoparasitic. So many boletes, most of the boletes all around the world, probably 99% of boletes are mycorrhizal, but this bolete is one that is saprophytic. So that means it breaks down dead wood, breaks down dead plant material. And this comes just after a week or two after I got the Phlebobis portentus. I don't remember how you say the last word, but the Mushroom Shop LLC, Jacob, sent me along with that Mongolian Lion's Mane a really cool bolete, which this plate's got some condensation. The original plate I saved it just in case, but you can see like that is crazy. That's a bolete. So this is a different cultivatable bolete, right? And so it's just taken up on. It's just I'm excited. That's the odds of it. So it's just taken up on agar. It's the leaves a little color. It's weird. It's a weird little specimen. So I just got that bolete from Jacob. So now I've been on the lookout, thinking about boletes, and was curious about different boletes that you could maybe cultivate. And so, actually, just on a walk a couple days later, I saw a handful of these, and they just the color and everything, something about them. I was like, these are cool. Like, I try and ID a bunch of specimens around me, but sometimes they really want you to know who they are. They just like pull at your strings at your brain, and you have to figure it out. And so I had to figure out what culture this was, what species this was, and I'm learning a bunch of others too. But this one, Buchwaldo boletus hemicrisis, saprophytic mycoparasitic. So breaks down the dead plant material, but also nearby there was a bunch of velvet top polypores. I've been seeing velvet top polypores for a couple weeks on this trail. I'm like, I mean, those are cool, but like, I don't know, they don't really get me that excited. They kind of look pretty rough, especially in all the heat and some of the dryness. They're not pretty looking mushrooms. And I'm like, they're cool, but I don't know. I think those are mycorrhizal or something, or maybe not mycorrhizal, but they need the pine. I don't know. Nothing about the velvet top polypore got me that excited. Maybe they're awesome. We should probably save some of those for us too. That's what this is all about. But this bad boy is a really cool cultivatable bolete. So something about it just struck me, and I needed to figure out who it was. And so I just brought one of the mushrooms, nice plump mushroom, back home. And you can't see it because it's black. I gotta work on my lighting. This is ridiculous. But it's just starting to fuzz out. That's the Buchwaldo boletus hemicrisis. Got a couple of the plates, but that one looks like the best one. So that one just from the other day. And so what I do is just take that mushroom, break it right open, and take a nice clean piece of flesh from the center. Put it on an agar plate. That's a carbon water agar, or charcoal water agar. So instead of just no nutrients and just agar and water, I added a little bit of carbon, just to help kind of hold some things back a little bit, but also give a good contrast and color. It kind of helps with bacteria and stuff, some contaminants the carbon does or the charcoal does, but I wouldn't count on it. But so that's basically just a water agar. But yeah, so it's cultivatable bolete. Right after I got a different cultivatable bolete from the Mushroom Shop LLC. Shout out Jacob. So I'm excited. Now I got two different cultivatable boletes to work around with, and just get weird, get wild. But this one I found myself. I found out in the wild, growing in the heat. Again, the cicadas have been ripping every single day. There's a dude with a harmonica behind me at all times if I'm outside, because it's hot, but it's nice. I'd rather be hot than freezing in Maine winters. But I found this one outside recently, so I'm excited. I'm going to try and cultivate it. Speak of the devil, also, the Jacob Mushroom at the Mushroom Shop LLC. I saw him post today. He found one of these, or maybe Buchwaldo boletus lignicolor, possibly. It was a little more brown, which technically also there's, I think, literature. There's a little bit of literature that this one, the hemicrisis, is mycoparasitic, but most of the literature is that the Buchwaldo boletus lignicolor is mycoparasitic, but this one displays it, and also looking around in its ecology, it seems to confirm it, from not just myself, but other reports online. Seems like other people are. So So that's also cool, right? And so not only am I saving and preserving these wild genetics from this local area, proven to grow in the heat in August in Florida, while like, there's not much that likes to grow in August in Florida, but this mushroom loves it. So while it's not going to be a heavy yielder, it gives potential that in the future I could grow it and get yields this time of year. But then it's also because I'm interested in it and something about it calls out to me in the color, and I'm looking into it and I'm figuring out, oh, it's actually mycoparasitic. And so then because I'm working with and trying to collect and learn more about this wild specimen, I'm learning more about its ecology. So I'm learning more about the velvet top polypore and then the pine trees that they associate with, and this whole new level and depth of understanding and just even observing the natural area that I'm going to just starts to develop as I continue to get and take these clones and have these cultures. Just like deepens the relationship and the knowledge and the just connection that you have to these woods, to the areas you're getting these clones from, especially if you're able to collect them yourself. Getting wild clones and like sharing wild clones with friends and things, like it's, you can get really excited and feel good, and like there's the connection to them. But like I mean, as somebody that's not a parent, right, I'm not a parent, but it's like having a baby, but like you just found the baby, you didn't have it, you didn't have to go through the process, you just found the baby and you get to enjoy it. uh, but it's like a pet person, you get connected to it, and like you remember the day that you went out there and you turned the corner and you saw that big lion's mane, or you turned the corner and you saw your Buchwaldo boletus hemicrisis, or you saw your aspen oyster, whatever it was. You can remember the times that you found those mushrooms. It's just like eating it leaves that like kind of memory in your brain, because it's like your body is a forager's, like this is where food is. And so at the same time, when you pick them and you can clone them and continue to grow them out and then produce pounds and pounds and pounds of food, and also be able to save and preserve those genetics, your brain is like, yes, I remember that. It's like a key moment, at least for me. I don't know. Maybe that's just me. Maybe other people clone mushrooms that they find in the wild and they're just like, whatever, next. But I love it. It's like it forms this deeper connection to your ecology, your local ecology, your local ecosystems, and all of the different connections, because you just want to learn. And then, like in this case, I need to learn how to cultivate this bolete. So I'm gonna have to learn way more about what I'm seeing and finding in these pine palmy flatlands, and how I can kind of recreate what this culture likes. So then I'm gonna have to understand more and more about forest. And so then it's, as I'm collecting these wild species, I'm just developing more and more and more as an ecologist and conservationist, even not even just a mycologist. And so this one, I just kind of mentioned it, but I forgot I didn't actually put it up here. This is my first clone ever. I 2023. I think it was like June 3rd. I think June 3rd, maybe June 2nd, 2023. I saw it for a couple days and I let it develop at this old growth trail. Yes, 1000%. This is why we find and clone local things. And so I'm in Florida now, but I lived in Maine, so that's why I've got some Maine cultures. But that's one of the biggest things is also like, just get out there, look around, enjoy yourself, make friends with the local trees, the local plants. Go in a couple weeks later, check on them, maybe next day. But like, you get to kind of go through the seasons and see how things develop. And like the case of the aspen oyster, I didn't see it pop up again, but the bear's tooth and the coralloides, when I was going to this Maine old growth forest, or not the coralloides, just the bear's head. Sorry, but I saw it fruiting in 2023, in August of 23, and then that same log and spread further. I saw it in 24, and then unfortunately I wasn't there to check it out in 2025. But I'm hoping next year I can get back up to Maine so I can check it out, because it's just through the seasons, through the years, you get to, yeah, especially as you're looking for mushrooms, and trying to just, whether it's for cultivation or just to learn kind of what the mushrooms are in your area. Um, you just develop that relationship, and like through the seasons you get to see the trees change, you get to see how much different things grow and develop, and just you become friends with your forest. As cheesy as it may sound, maybe to other people, if you know, you fully understand what I'm saying, and you just, those daily interactions, walking through looking for mushrooms, uh, even if you don't find any, find any that you can eat. They're just super impactful, just as a person, as a human. So get outside, find some oysters. So this one was my first clone ever, my first wild clone. I think I need to check my phone. I've got so many mushroom pictures and videos and things that I don't know if I have a video or pictures of cloning this one. It'd be really cool if I did, because yeah, it's the first clone I took, the first wild specimen I took. I started growing mushrooms and diving into the mushroom rabbit hole early January of 2023. My friend gave me a lion's mane kit from North Spore for Christmas. Let it sit in the closet for like a week, 2 weeks, because I was just like, I don't even know what this thing is. And then also at that time, I got some mushrooms, other mushrooms. And then after that, I was like, you know what? These things are kind of interesting. And that opened up a whole wormhole. And so then 6 months later, I found myself in the old growth woods cloning some aspen oysters, Pleurodus populinus. And so that's what it looks like, bottom left. I've got some pictures I've shown off on Instagram as well. There's a bunch of bugs in it and stuff, but not too buggy. But so I just grabbed a couple little oysters off of it, brought it home, cloned it. You can see compared to some other oysters, it's a little thinner, and it almost always has like a couple little segments on it compared to some of the other oysters. It just doesn't. I've put it on like water agar and different things, and I've been like, maybe it's got a little friend hang on. It's always done it, and it's always then still produced. I've been able to grow it probably like mid 70s, almost like 80 degrees here in Florida, but it didn't really do well, and it got a little blotched out. But it's got this really, really beautiful. It didn't really, like on the clone itself, the wild specimen, it was nice. It's just like a pearly, off-white, almost little cream oyster, right? But then once you grow it, and I've got better pictures online that you can see if you look up and look on Instagram and such, but it's got like this really, really nice marbling on it. And so it comes in like this lighter cream, almost off, look just slightly off white, and then streaks all throughout of just like this darker, creamier, just marbling. And it's beautiful, and it's got a really, really nice kind of like black licorice. There's like a few different, I don't know what it is, I don't know what the black licorice scent is. Is that like maybe anise, right? I know anise is like a common mushroom scent, and some of the mushroom smells and names and things don't make sense. Like there's like ferris or something is like cucumber, I think is like feraceous. Is like a cucumber scent. It's like to say it's cucumber scented. I don't know. But so this one's got a really, really sweet smell. All oysters have that oystery smell. But this one's I don't know, I need to learn my smells beyond where they're at. But it's something about some of them just stick out more than others. It's like one of them in terms of like actives, like I know like Melmac. And those varieties smell so strongly of feet. Like they got the most funk and like rich, earthy something funk to the smells. And so this one's on the different end. And it's kind of like a nice like black licorice anise oyster of sorts. And yeah, it produces pretty well on Masters Mix and such. That's a 5-pound block. But it's not quite as not quite as resilient and variable as in temperatures as some of the other lion's mane and stuff I was talking about earlier, but still a beautiful wild culture. My first culture, and again, it's from an untouched old growth forest in Maine, so it hasn't kind of been too affected by different lumber and farms and urban sprawl and pollution or anything. It's fairly well wild-ish, not wild, but wild, right? Compared to something maybe like the brick house lion's mane, which is still a great wild specimen, but maybe that one has been selected a little bit more by some of the urban pressures, which has its perks and uses, especially here in Jacksonville. But then there's also, something to be said for something that's maybe left a little bit more pure in a privately owned old growth forest, right? There's value in both of those to see what's in those genetics, especially as we move in the future and learn and understand more and more about kind of these different compounds, the DNA, the different species. Why wouldn't we just be trying to save all of them, basically? So that's the vast majority of what I've grown, right? That's where most of my experience on this is going to come from, is just those handful of cultures that I've been working. For a while, I was working and focusing with more proven genetics, more commercial cultures. I signed up. It's a great thing. I signed up for the culture club with Myco Wizards, main cap and stem. Got a bunch of cultures that way, a lot of commercial cultures and proven commercial varieties that way. And I run a bunch of them still, right? Like there's a place for them. It's not like I'm saying don't run your commercial varieties, only run the wilds, because truth is sometimes you go out in the woods and you grab a lion's mane clone or something and it just sucks, right? Like it just isn't, it's weak or it produces thin. They're not all the same, but there's value in there and there's different genetics. And so just like you don't want to take one family, right? You look at some inbred families and people and animals, and they're a little messed up, and it's because they've been so like localized and trapped in the genetics in this one little loop, without being able to escape. And so being able to kind of preserve and have all these wild genetics from Maine, from Virginia, from Florida, from here, from there. You're having these totally different gene pools that you're able to mix in to these different cultures, species over time, that maybe you're seeing some of these issues with. So maybe some of these, like I know there's like zoos and stuff that preserve a bunch of like tissues, like different cells and tissue lines and DNA from a bunch of different animals and mammals. So then in the future, when like there's certain diseases and pests taking out a bunch of a certain species, like a bunch of a certain animal, and the gene pool of this almost what's it called? Not many of the animals. I'm not a zoologist, but almost extinct. That's the word. So when there's a bunch of these animals that are going extinct and there's just not as much of a population, you're going to see the gene pool and the population size just continue to shrink, and then different pests, different diseases, different deficiencies, different things like that are going to come forward and present themselves more so because they just don't have the genes to defend themselves, or maybe with all of that inbreeding, you're kind of selecting for some of those recessive genes that aren't as desirable. But then being able to have these wild genetics, wild DNA, kind of from out there, from different areas, from these untouched sources, we're able to then expand out and bring in this like nice F1 vigor, this whole new gene pool, whole new set of possibilities, and then be able to have those animals bring in, like in the zoos, they'll freeze DNA from an animal from like the 80s or something. Luckily, they were doing it, and so today they'll have issues with pest and disease, or different things, different diseases and things taking out a specific animal and that small population, and so then they can reclone and like bring this old animal DNA back into this new population. And produce new offspring via cloning. So we're doing the same thing with mushrooms. The zoos have to have really fancy technology and crazy things to do it. We can do it in a bedroom, in the closet, in a lab, in a tent, in your kitchen. You can do it anywhere. So this is just what I've been growing. I haven't mentioned it yet. iNaturalist. Use iNaturalist. It's gonna be your best friend. Uh, So iNaturalist, I've been using it a lot, especially down here in Florida, just trying to learn what fruits in the area, what I can expect to see, what I can expect to find, and just in the seasons. To maybe this time of year, you can select to look on iNaturalist, like what people have found in September in this area last year and previous years. So then I can kind of see what might pop up in September in previous years. And so iNaturalist is an app you can it's like collecting digital Pokemon, right? You can go, you don't physically keep and use there's people that have to actually, if you're going to clone it, you have to remove the mushroom. Sometimes, not always, you can do some needle biopsies and things. But then there's a lot of times people will just take pictures of mushrooms. I'm not going to clone like every single amanita and stuff. And there's a bunch of things that I'm not sure if I can grow it out. So I'm not going to just clone everything. Sometimes I just want to learn what they are. And so iNaturalist is a great app for that. You can use it on your computer, but it's great on your phone. So then I'll just go out, I'll take a whole bunch of pictures of all the different mushrooms I see. Or you can do it for plants, for animals, for flowers, trees, and it'll generate. It's got enough data in there. It will use AI and some different systems to then generate what it thinks the species is. Sometimes it just says it's a guild mushroom, and that's the best it's got for you. But sometimes it can do pretty well. It's not always accurate, so I wouldn't rely on it. But in using it, you're going to continue to improve its accuracy. And then also, you're going to continue to provide a kind of a database for yourself to even continue to refer back to and figure out where you found certain species, what time of year. And as you continue to collect them and you see some different repeats and some different consistencies, and maybe it's always after 3 weeks after it pours or after this temperature hits this range that you're seeing these different boletes or this different thing pop up. So it's a way that you can continue to connect to your ecosystem, your local ecosystem, and then even use other people's data to do so. So even though you haven't seen every single mushroom and specimen and bug, and not every single one that people see, they're going to post on iNaturalist, but it still gives you a really good idea of what all is going on in your area. So like this little map here is just mushrooms, and I don't know if it's how long it goes back, but that's just mushrooms in Jacksonville. All those little spots are different Jacksonville mushroom sightings. So depending on where you're at, maybe more people use it, maybe less people use it, but still it provides a little bit of a background and some information on what you can expect to find in some local areas. And you can get some IDs yourself if you post them. It's just like that nice database that we can all refer to and refer back to. So even if you take clones and you run those genetics in the future, blah, 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 you have your iNaturalist ID you can go back to and see the exact day, spot. You can move the spot if you don't want to give up your honey hole, right? You don't have to give the exact location. You can put it like 300 feet up trail or something. But it's really helpful to then just provide some data behind the finds, behind what's out there in the woods, what you're finding, what other people are finding, and just get better connected and learn more about your local ecosystem. So I've talked a little bit about it in going terms, a little bit over kind of why and like each of these specimens. So this is just a little rundown on it. Again, one of the biggest reasons why preserving and cloning and working with different wild specimen, like I was saying with the black marsh lion's mane, which really sparked this whole thing, this whole live topic, is the beach leaf disease. And so there's a bunch of different pests and disease, and I haven't listed all of them, but these are some of the major factors and things that we're seeing issues with. And so there's the beach leaf disease, which is just coming through like nematodes and water and spreading leaf to leaf, and just wiping out a whole bunch of the beaches in that. in that Maryland area. And then we see like a whole bunch that's common on the East Coast. Where I was living previously, I had a tree in the front yard impacted by it. And so like the emerald ash borer, that's a known one that's going around everywhere, wiping out a whole bunch of ash trees, which things like different conchs, hericiums, and oysters love to grow on. So we're going to continue as those continue to proliferate and then spread. We're seeing a decline there. White pine weevil is another one, especially for some really prized specimen and unknown species. Right, like we're still learning a lot on like matsutakis and how we're not cultivating them, but we're still learning a lot and trying to understand matsutakis and way more about them. And so as the pines are kind of threatened here and there, and as the white pine weevil and things like that continue to spread, we're continuing to lose data and ideas and genetics from different areas. And then there's also different things like invasive fungi and things like oysters or the golden oysters and things that are popping up, and even like honey mushrooms, and those are able to outcompete maybe some more native fungi and some things that are more a little slower but more in the area. And then things like the oysters or the honey mushrooms are going to come in, outcompete them, and kind of take over that niche. And then we also look at just humans, right? Like, as all the things that we're doing, and that's really enough on all the reasons why we need to preserve it, and work to save any of these genetics, especially the ones, obviously, like every mushroom has its purpose, right? Like if I bring some clones sometimes, and I'll have some mushrooms, and there'll be people, friends, family, that whatever, that ask me like, oh, well, what is that? Like, what's it good for? Kind of a loaded question, and it's hard because everything's good for something, right? Like, maybe not us. We don't need to use it for anything. Maybe there's uses for it, but we just don't know them. But that doesn't need to always be oh, well, this one you can turn into a cream, and if you rub it on your ass, then it makes you really strong and healthy. Or this one you put up between your toes, it makes your feet smell good. I don't know. It's good because it's cool. Like, what is it good for? It's good for growing itself and being itself. I don't know. All right, we're still learning so much, but especially with these specimens that we know are impactful and are already in society and being used for a bunch of different things, like these different reishi or these different lion's mane and hericiums or oysters, or maybe like that bolete and some different things like that. We're seeing a bunch of issues from humans. So us as humans should probably work to preserve them a little bit. And it's things like deforestation, pollution, climate change, and even just like overall degradation of quality, right? And so even if we're not doing full deforestation, we're taking some trees or replacing local areas where we did take a bunch of trees with just a whole bunch of monocrops planted in uneven, unnatural positions and rows. And so that's kind of helping different pests and diseases and issues proliferate a little bit easier in those unnatural conditions, compared to what would kind of naturally keep them in check. So there's way more reasons why, but these are just some of the reasons. Because we're dirty, we're raising and changing the climates, we're loud, we take all the trees, and then we put the bad ones back in proper places if we put anything back, so then we're just throwing off the whole balance of ecosystems and nature and ruining our fungal flushes for years to come, without even realizing it. So hell yeah, this guy. So anybody that ever sees this at any time, if you want anything to have in your life to taste delicious, it's this guy. Go to red flower fungi, get some seasonings, get some flavors, get some ferments, get some koji, some Leoncito Cafecito, some good lion's mane coffee. This guy's got it. So welcome. Thank you. So yeah, really, whenever we're working with these wild genetics and wild specimen, what we're doing is just kind of helping improve our own resiliency and diversity, and doing so by the selections by the really the greatest breeder, right? There's a lot of mushroom breeders and like pepper breeders and potato breeders and cannabis breeders and things, but the real breeder, really best one out there, top notch selections always is going to be Mother Nature, right? What makes it in her climate without the assistance of us constantly watering and feeding and pruning and this and that, whatever it may be, is really the best selections there are. So that got a little shifted. That's fine. But yeah, they might not always be around, right? They're going quickly, especially here in Jacksonville. I look around and there's a whole bunch of places that I remember as a kid just being woods and trees and swampy areas, and my dad even saying, like, even this time last year, these places were trees and woods and swampy areas, and there's just everywhere I look, there's just clear cuts, bulldozers, excavators, new apartments, condos, just shopping malls, not shopping malls, but plazas. The malls are dead, but just there's so many new things constantly going up, and this little bit of green space, the tiny little bits of trees and everything are just gone, just like that, like so fast. And I just saw a new thing that there's a big plot of land and forest somewhere here in Jacksonville, and I guess they're doing something for developers and things to speed it along even more so. So we're going to continue to lose even the little bits of land and things that we do have out here, the little bits of green space amongst all this concrete. So we don't know how long we're going to have it, right? But we also see things like in DC and like Microdex. It's what some of his great work, and he's got a bunch of really cool wild specimen. He'll just be walking down the sidewalk, and just some of those casual oak trees and different trees, and little areas along the sidewalk. Really, if you're looking for them, they'll find you and you'll find them, right? And so that's even amongst all the urban sprawl and concrete. We'll keep looking, and we'll try and find those proven wild specimen, those ones like those trees you see growing out of gutters and off of roofs. We'll just have to find the lion's manes and the oysters that are growing off of buildings and things. Probably oysters. They will probably grow off of brick buildings before we know it. So yeah. Find some local woods, get out there, find the local backyard trees, a little bit of greenery, a little bit of woods, the older trees, the older an ecosystem and area has been around, you'll see way more diversity than if it's a pre-planted forest or planned out like just a whole bunch of rows of pine trees or something like that, compared to if you see something older and a couple different species, you're always going to see more mushrooms in those areas. But you got to look for them, and they're out there. Really, fungi are quite literally everywhere. I think there's even some in like Antarctica, and if you look at like lichen can survive in space, but lichen are half fungi, half bacteria. So I mean, they're everywhere. So go to your local woods, look in some sidewalks. Check your backyard underneath some areas, woody areas for them, depending on the mushrooms, and like train wreckers like grow down here, and those can grow on like pressurized treated pine and things like that, and like railroad ties. So like they don't really care. Like if there's conditions and you give them a chance, they'll grow. And then preserve it, right? Preserve it, document it, take notes, take pictures, put them on iNaturalist, take spore prints yourself if it's maybe depending on your local regulations and rules. Like I know in some spots in Jacksonville you're not supposed to take any mushrooms or whatever, pick anything or any plants, but it's for the greater good of society. So if somebody wants to come yell at me, I mean, fucking do it. I don't know. I'm trying to teach people about mushrooms and get them more involved in mushrooms. So if you get mad at me for plucking a couple mushrooms out of a park in Jacksonville, then I don't know. I don't really care. But I'm going to keep looking for them, and I'm going to keep documenting them. So I'm going to keep uploading them. I've got a bunch I need to get on iNaturalist. But I'm trying to I want to be able to identify them, and I don't like when I put them on iNaturalist and it just says guild mushroom. And eventually other people can come in and add some comments. But this is where it's also, I didn't mention it, but if you're on Facebook, I would, even if you don't have Facebook, get on Facebook, even just for some of the local mushroom communities and things like that. I've found in Maine that was one of the best ways for me to learn the mushrooms, and I'm still learning a bunch down here in Florida, and it's not as lively on the Florida mushroom pages as some of the Maine stuff. But it's a great place to go to Facebook, look for your local mushroom, your state mushroom groups, your local mushroom groups. They often have identification groups and things in there as well. So like that Butchwaldo boletus hemicrisis, I didn't know what it was. Nothing on iNaturalist was coming up, and I didn't find it in the book. It was actually in the book. I just kept missing the page. But I couldn't figure out what that was. And so like that and some other specimen I've posted in some Facebook groups, and there's always within probably an hour, 30 minutes. Sometimes it's slow, but often there's somebody, whether it's a nice young person or some 80-year-old person that's just been there, done that, seen the mushrooms in that area. Not always right. Double check what they tell you. But a lot of times those local mushroom pages on Facebook and things like that can give you a whole lot of information and help you, even if you just scroll through and see what other people are posting. You can see a bunch of different things, learn what maybe you might see out and about, and what other people have going. So yeah, we're going out there, document it, take some pictures, get involved locally, right? And so there's a bunch of different ways, like even just with all the stuff going on in Jacksonville, I've been pondering and looking into some different ways that I can get a little bit more involved locally in saving and preserving some of these areas. And I think part of that also can come down to understanding more of the fungi that we have down here in some of the mushrooms and the potential, and just the overall fungi, not just the mushrooms. I think understanding some of the importance of some of the diversity of species and some of the species in specifics can help with some of the constant loss of lands and things that we're seeing. Is just being able to, unfortunately, figure out how to place a value, which is gross because it's nature, and like it doesn't need to have like an economic value. But I think also people in like Jacksonville and some urban areas don't understand the value in ecosystem services and different things that these wild areas, and diverse wild areas, are able to produce and help and provide for us. So I feel like sometimes that maybe I should get a little bit more involved in some of the local issues like that. But then maybe if I can just lead locally and get more troops on the ground, maybe that's where my time is better spent than yelling at people in suits indoors, doing politician things. That's not really my style. So I'm just gonna be here on the ground, getting people going, and other people do it. And other people will be making moves in the mountains. I love it. Swamp style, does that mean you're going nice and low tech? I like it. So yeah, and also spread local spores, right? Like if you find a local lion's mane or local oysters or a local funky active mushroom or whatever spores, local specimen you're finding, collect some mushrooms and spread those around, right? Like it's already proven in your local area. Don't spread gold oyster freely around just cause, I guess. But like spread the local native fungus you find, collect the spores, make liquid culture, make grain spawn, whatever it is, spread that around. It's proven to fruit in your area already. So by helping that produce a little bit more, you can set yourself up with kind of just some freebie fruits and some freebie mushrooms down the road, or at least be able to spread it. Like I know people take their wood chips and their spawn, like in the northwest of some different like ovoids and things, and add those like water balloons and such, and add those like police stations and whatnot. You can do that same thing with reishi and lion's mane and oyster mushrooms and boletes, and spread even like morels and hope for the best on that. Chanterelle, like try and do all this different stuff and spread these local specimen around, especially with it being, if it's a local collection, like you're not hurting anything. Don't even, I mean, I'm growing wilds from Maryland and Virginia outdoors. So like, don't sweat it, especially at a smaller scale. Like, don't sweat it too much. But local is definitely best. Cloning, I won't go super hard deep into cloning. We'll do a whole videos on that, and I'll post videos when I can do it when it's not live streamed. So that little video of myself is a little bit clearer and nicer. But basically, what I do for cloning is I'll take the fresh mushroom, depending on what the species is and how I've collected it. I'll either get prints from one or two of the species, or sometimes I'll get prints after I get the clone. It all depends on the shape of the mushroom and how many you have to be able to select from. But like previously, you see right there, that's one of the Ganoderma courtesies. And so that one's kind of a tough clone. Really, for some Ganodermas, if you look at the real base of it, you can get some chunks of wood sometimes. And so that makes it easy if you can get a piece of the wood that's already growing on. You can get a good clone off of that. Spores are a little bit harder off of some of these smaller Ganodermas. There's ways to do it, I'm sure, but I haven't had that good success of just leaving them for a spore print. Maybe if I could leave them in the wild and like leave them connected and put like, I don't know. Maybe it's something I'm doing wrong, but I haven't had as much luck with the spore prints. But if you can go from the base of the Ganoderma or right at the gills, if you can get a good clean tear without squishing too much, if you squish, you're kind of just helping spread any of the water and bacteria that's maybe somewhere in the mushroom. You're spreading that all around. So you want to tear into the mushroom and to kind of reveal that inner fresh, untouched by the outside air flesh without squishing and mushing it up too much. So rishis, it can be kind of hard to do it. But often I'll use either water agar or, like I showed earlier, just a carbon agar or charcoal agar. So that's just using the agar for the solidification and water. So no nutrients. The charcoal is mostly just for color, but it has some potentially helps with some different bacteria and things for some contamination. But really, water agar is best, or low nutrient agar, so you can drop the nutrients down a little bit. I try and always have a handful of those plates kind of available. I don't want to pour them whenever I find something cool. So whenever I have to pour plates, I'll just make 1 liter that's just plain water agar or just charcoal agar. So then for a little while, whenever I find wild specimen, I can bring them home, bring them from the flow hood, and then I'll literally take it, rip it center open, leave the center facing the flow, so all the clean, fresh air is just coming right to the center flesh, and then you just tweezer out with. Every good mycologist, right? Every good mycologist has a good collection of tweezers, and there's a scalpel in there, but these are mostly all tweezers, and those aren't even my good ones. My good ones are in there. Right. Yeah, tweezers for days. Some of them are like some little needle point. Some of them are a little thicker. Right, and got a little something to them. This one's like a nice flat. Get yourself some tweezers and get yourself some different the needle point's the best one, but this one is nice, the flat, sometimes just to get yourself like a little bit of meat. But get yourself some tweezers, right? Every good mycologist is going to have a couple sets of tweezers. And some of them are a little nicer than others, and some of them bend and deform over time. But it's to be able to then rip that fresh mushroom open and use those tweezers to get a nice little piece. You can cut it out. You can use the scalpel and cut out like a little square and pluck that little piece out, especially for softer mushrooms. That's pretty easy. But then at the same time, the tweezer, and you can just like pluck like a little string cheese piece of it off. And I found that it works. You have to have a stronger set of tweezers for the reishi to be able to rip off an inner piece of that reishi sometimes. Reishi's like 60% water. It's way more woody, way more chitin than a bunch of other mushrooms. So that one's a little tough, but usually those tweezers right in that center is critical, just to be able to get that quick, easy, clean clone off of it. And red flower, Thank you. I appreciate you. We gotta get together soon. Catch up, do some swapping of cultivation and cooking and fermenting ideas, and we'll have them on. We gotta have you on here soon so that we can just do a talk. One of these Mondays or something. We can hop on and just do a little catch up, a little chat, see what you're up to, see what I'm up to, and let's do a little check in. So I've thought about it. I need to do I think more. I've always wanted to kind of do a podcast in like 2024 when I was originally balls deep before life gave me a little speed bump in my mushroom journey. I wanted to roll out a podcast in 2024. So that's why I started just doing these live videos, really, is just because last year I was just in a cold room making a lot of hash rosin every day, and I didn't really get to do or talk mushrooms. Well, if that's the case for mushrooms, I'm bougie. All right, I got like 12 sets of tweezers, but they're crucial. They all have their own like perfect need, and sometimes I have to come in the lab because I've got a fucking splinter, and I'm like, no, I've got the perfect set of tweezers that'll get that out, and then I flame sterilize them, and so they're clean. But yeah, so I've thought about doing a little podcast or something. So maybe we gotta hop on the chat, Red Flower. Nick, you're great, and you got a lot of really good ideas that I know a lot of people would love to hear. So we'll get you on here, or do something. Maybe it doesn't have to be live, but we'll make something happen. But yeah, so that's one of the ways is I will often use just tweezers and that inner flesh, depending on the species. You'll see right there, and I talked a little bit about it earlier. Is that for lion's mane and some other species, I'll use like an isopropyl soaked mason jar ring to then be able to elevate the mane above the aluminum foil, so then the spores are able to drop off a little bit rather than just the gills sitting on the foil. And I've used it for like bigger oysters as well. I don't like the oysters just sitting there on the dish and the plate. So being able to use like the mason jar ring, I've also used like the tweezers or something or scalpels. I'll just set on the side, chopsticks. You can use all sorts of things. There's like a little riser just to keep the gills and the mushroom flesh up and off the foil a little bit, and then get some nice clean, fresh spores. And then you can take those spores, use a sterile cotton swab, and put those on some different agar medias. You can take some of the spores and a little bit of distilled sterile water and make a spore syringe, whatever it may be. Or you can just take those spores, fold them up nicely, label exactly what they are so you don't forget what they are as soon as you put it away, and then use them for long-term storage, depending on the species and the spores and all that. Some of them, like, maybe are only going to be good for a handful of years, but often a lot of them will be good for like 10, 20 years. You'll see a decrease in their viability kind of over time, just like with anything with seeds. But they can stay for quite a long time. So I've even seen people bring, I think, spores to space and bring them back down, and they didn't really do anything. So then you can always come back to them. You can take spores, these lion's mane spores. It feels good because then I'm like, okay, if I were to drop something and break all of my petri dishes, and then the fridge gave out all on the same day, so then all of my the fridge giving out wouldn't ruin it, my long-term water vials. But the thought of losing my water vials and losing my cultures or something is scary. But then if I have a bunch of spores backed up, which are just this low-tech little sleeve of aluminum foil, you can put them all in like a different binder, a different folder, in a book, in a bin, whatever it is. And then all of these are wrapped in aluminum foil. And then I'll put them in a little plastic baggie, and then they're in another container. So I'm like, I feel good once I have spores backed up. I've got water vials, and I've got a couple plates sitting around of a culture. I'm like, this thing ain't going anywhere. Like to lose all of those, right, is crazy. So that's why I'm also ridiculous and I keep too many cultures. I have an issue, a good issue. But then yeah, with the prints, with the water agar, depending on what your culture is, maybe you've got a really wet culture or a really wet specimen, and it's hard, it's a little bit more bacterial, or you're having some issues with it. That's when you want to go to prints, go to spores. And spores can definitely be dirty, especially coming from outdoors. You can see a bunch of contamination. So you kind of got to know what to look for a little bit and be patient. And again, that's where you can use some low nutrient agar to kind of help with some of that germination and reducing the contamination. But save the genetics, right? It's whatever it is. You can even use like cardboard, right? You can use like cardboard wine caps and things, and different like oysters. You can save the butt ends of a lot of oysters and just kind of throw them into a pile together, and you can kind of do a quick and dirty spread that way. Wine caps do that pretty well. But even like cardboard, you can use like some wet or pasteurized cardboard. I dropped my mason jar. If you could hear that banging around. So you can use cardboard, agar, spores, and then yeah, once I have my culture ripping, looking nice on agar, right? Like once my lion's mane's looking good on agar, I'd come in here and I'll grab a couple little sections and then put those in a water vial. And so I'll use 5 to 10 to 15 milliliter centrifuge tubes, and I'll fill them with some water, distilled water, pressure cook that like I'm cooking liquid culture for like 25 minutes at 15 psi. And then you've got a nice sterile water vial that you can drop a couple pieces of agar into. I think it does best technically with water agar or a lower nutrient agar. So that way the nutrients don't leach into the water a little bit and your mycelium just kind of goes into that quicker, easier dormant stage. But even so, it's as long as it's not like a super high nutrient agar, I haven't had any issues, at least storing for 2 to 3 years so far. I haven't been growing long enough and haven't been storing cultures long enough to have, decades of experience and be like, oh, I just pulled this culture I put in here in the 80s because I wasn't even alive. I wasn't alive till 98, right? So I can only do so much with so much time. I didn't know what mushrooms were. I did, but I didn't start growing mushrooms until think about mushrooms until the end of 2022, right? I worked for Northport for a day and I still didn't even know what the hell a mushroom was. And that was like 2021. It's part of my favorite, my villain origin story or something. I don't know, my backstory. Could make it sound more interesting. But I did work at Northport for a day in like 2021 before I knew anything about mushrooms. I don't remember who it was, but a very nice looking lady in the lab gave me a bunch of mushrooms, a bunch of pink and gold oysters. She was telling me all about them, but she was just very lovely, and I was lost in her eyes. So I didn't learn anything about mushrooms there. But then, yeah, I worked there for I think like a day, and then I ended up getting a job that was going to pay me more. They called me on like the way home. And a job nearby was going to pay me more for not as cool of work, but I had a senior dog life situation, whatever it was. So the mushrooms didn't win then. But then about 2 years later, I'm telling you, those spores got in, and I was just gonna be like in the back folding boxes. I wasn't gonna be doing like mushroom work. People always put me in the warehouse as like some brute worker, as like a skinny, frail little dude. I'm always just like giving warehouse work. It's crazy. So that's what I was gonna be doing at warehouse at Northport. I was gonna be working in the warehouse folding boxes like 2021. Didn't even know what a mushroom was. It's crazy. And now I'm here. Come a long ways. I'm glad I wasn't folding those boxes. But yeah, so water vials I haven't had forever to be able to grow them, right? I wasn't super into mushrooms until the end of 2022, start of 2023. I started diving into here, but even storing things for 2 to 3 years so far in less than ideal conditions, I've been able to pull a few things out of some water vials. and have pretty good success with the water vile. And if you look online, it's Castellini method, Castellani. I'm gonna have to look at that before I can even type it into the comments because I always spell it wrong. I'm not here for spelling. Yeah, I think it's there it is. So if you look up anything in regards to, yeah, see, you guys got the spelling. I wanted to make sure I didn't misspell it. All right, thank you. And I assume that's about me being lost in the North Spore lady's eyes or something. But I don't know. The first time seeing a mushroom lady hold mushrooms in your face and they're pinks and gold oysters, I don't know what they were. I changed my life, I will say. I didn't know it then. But yes, Castellani. There's some papers on him. I think he was like a French dude or something back in the day that came up with this method of storing agar. Or storing cultures in water vials, distilled water vials for long term, because it just kind of helps create, almost puts the mycelium in the cultures into like a hibernation. There's no extra food, there's no extra oxygen, so they kind of keep going for a little bit, and then the CO2 hits a point, and then there's nothing in the water, and they're just kind of like, all right, I guess we'll just wait. And then you can get to them like 25 years later, and I'm sure they're still just sitting there waiting, apparently. So that's a cool technique. So that's what I've been using. I've been taking a lot of my cultures and things I'm not running, even though I want to run and use all my cultures all at once. I'm just a one-man team, and I've only got one rack for my lab supplies and one rack for incubation. So I can only do so much. So the water vials and prints are a great way to be able to save cultures and specimen and not take up all the space and not have to constantly have those running and in use. And that was it, I guess. That's all I got. So if there's any specific questions, thoughts, I realize I'm just sitting in like half dark corner, but that's, lighting's poor. I don't really care. But yeah, any questions or anything, let me know. Other than that, we've run through all sorts of goodies. If you go on, look on like my Instagram, I've got a bunch of pictures of all of these, and each of them's tagged on who I got them from. So again, the black marsh lion's mane, one of my favorite cultures. Really, really great lion's mane, heat tolerant, from Myceliate. Really cool, small, workable little clusters on there. June cut lion's mane from Uncle Fungus. This thing's a beast. The most resilient lion's mane I've worked with in the heat. Yeah, right. So. Right, like there's like, especially with spores, like they stay from a while, and they for a very long time, and they're probably, like, I'm sure not every spore that was on that print, right, germinated like they would have maybe 10 years ago, but it just goes to show, especially for spores, like that's nature's way of storing these genetics. That's nature's long-term storage method, is just spores until it hits and it's able to kind of hit that substrate, or things get wet, or it finds its way into its next home. That's the beauty of spores. They're supposed to not germinate until they're in that right environment, right, where it's moist and nice and there's food. So being able to keep those spores on spore print, decade later, they'll still be ready. And yeah, in Maine, I had way more space, and I had like a whole basement to myself at one point, and like was almost about to get some things really running and booming. And then I got runned and boomed. But like, so now I just got one rack, and I mean, shit, I'll show it one. You can't see much. What is that? That bitch is. Let me see if I can loosen my camera. No, I can't. That's fine. There it is. I think it's full. All right, I got some cultures. I got some grains. I got some bins. I dropped my mic. But yeah, it's one rack's all you need, right? It's like a mason jar and a dream, or a one rack and a dream. It's all you need, the will to do it. So yeah, I can only do so much at once. I'd like to run more wild cultures, but that was the June cut. That thing's a beast. Virginia Lion's Mane. I got this from Performance Fungi, Lion's Mane James from Virginia. Medium heat tolerance, but this one's nice, sweet. I'm excited to have some spores off this one, run this one further, hit it to those other Lion's Manes. Brick House Lion's Mane. This one a little bit slower, but I think also shows partially how cool and how long some of these cultures can live. Like I was saying, up to like the possibly 40 years. But seen fruiting for up to 20, just hypothesized possibly Lion's Mane can live up to like 40 years. So maybe this one's an old culture. Who knows? I got to run it back and get spores. But again, just one dude, one rack. Maine Bear's Head. This one, it's end of August, right? It's end of August, not coming in on winter yet. But winter in Florida, I'm going to bust this one out and hopefully work with this a little bit. So anyone else with Bear's Head, let me know so I can work with those together. Especially if anyone watching this, if you're in the Florida southern area and you got some good wild clones, we got trade. Ganoderma Courtesy, this one hitting grain this week, so excited about this one. First Florida clone. Buchwaldo Boletus Hemicrisis, the one that just rolls off the tongue. Sounds like a terrible disease on one of those commercials about a bunch of prescription medication and a nice jingle behind it. But yeah, it's mycoparasitic, so it eats a velvet polypore, and it's also saprophytic, so I can supposedly grow it. Stay tuned, we'll see. Maine Aspen Oyster, my first clone. What started it all? Early June 2023. This bad boy. It's a little weird for an oyster, and it's not like as aggressive as the Florida oyster. It doesn't like the heat as much, but it's a beautiful, unique specimen. I'm really excited to have it backed up and have the spores and be able to continue to work it further and have these cool old growth genetics untouched by a lot of man-made influences and be able to have them down here in Florida and see what they can even do in Florida on the opposite side of the United States, where they probably would have never ended up in nature. But maybe they'll turn invasive. I don't think so. I'm not worried about it. So yeah, that's what we got. iNaturalist. Download it. Yeah. Thank you. I appreciate you all for coming in, listening to me talk about random wild mushrooms and just babble about half-related topics. The next week we'll do something. I haven't decided on the topic. Maybe Nick, maybe we'll catch up. I'll reach out to you, see what you're up to. Maybe we can do a little catch-up episode. Maybe I'll just do. What do you got? Yeah. So so I saw on Instagram, I can't think of his name. He's, I believe, also somewhere in South America, maybe, or Central America. I might be making this up. He's somewhere, I believe. He speaks Spanish, I believe. And he's a really cool Instagram account I follow. And recently he's posted a few things, and he posted a nice video and reel about this specifically and the using the filter paper. And I think that's also part of, like, it's a good hardy structure rather than like the agar. As it sits in the water for 10, 20, 30 years, is going to want to slowly break apart. So I've always used like a thicker agar when I do it. I try and use like a nice stiff, at least 18 grams of the Gelidium agar. So it's a good snappy agar. But I think the people use the filter dish, and part of the filter paper and filter disc is that it's able to just maintain itself a little bit easier structurally as it sits in water for 10, 20 years. And there's not a bunch of nutrition all over it, so it's not the agar leaching the nutrition and almost creating a little vial of like a little liquid culture vial if it breaks down enough. The filter paper just kind of hangs out, right? So I think that's part of why the filter paper is great. I haven't done it specifically myself. I did mean to actually mention the filter paper specific, so I'm glad that you brought that up. I meant to add that with all of that talk about some of the Castellini method. So yeah, look up Castellini method. Use agar and distilled water, or you can also use the filter paper and distilled water. I do plan on actually going back through some of my cultures and doing the filter paper method, even as just a secondary option. And then there's also people in places like Maine Cap and Stem that have access to like cryo freezers and things, and they take like mineral water and slants and like a little bit of colonized grain, put it in like a cryo tube, and used a little bit of like glycerin around the grain so the grain and the mycelium doesn't like fully burst while free. But I don't have a cryo friggin' freezer, and I don't think you do either. So filter paper and water bottle till then. But yeah, that's it. Any questions that come up, reach out to me, comment on here, reach out to me on the many platforms you can find me on. Like this video, subscribe to my page if you haven't. If you're here, you probably already at least subscribed. But hit that like button. Come on, do your boy a solid here. Hit the little like button or something for me. Play the little game for me. So we can keep talking about mushrooms and keep reaching more people and keep babbling. And again, I'll get some people on here. We're going to have a bunch of more ideas. This is just a continuously ongoing Monday meeting, a little myceliation on Monday. I used to love a couple of years ago being able to watch like the Myco Geeky show on Monday to kind of start your week with some. It's Monday. It starts with them. We got to have something with some mushrooms in there. And that's not going anymore. And so someone's got to talk about mushrooms to start the week, right? Mossy Creek does his Thursday live streams, so Mondays are open, and we're here in the swamp. So reach out, check me out, Instagram. That's where I do most of my posts and show off most things for a while, but I'm trying to do more and switch over here to more YouTube, some of that more long-term content, and reach more communities. And so I'm also on Patreon, where you can subscribe, pay and get a little extra content. Subscribe for free. And so we got a lot of really good information that I'm not sharing on Instagram or anywhere else. And as always, our Discord. The link is on my website, on Instagram, and on Patreon. But if you still need assistance, hit me up, and I'll send you the link for any of them for our Discord. Join in our communities. Keep learning. Stay swampy. Thank you. I'll see you next week. Thank you, guys. I mean it.